right, thank you all for the opportunity to share some of my work. I'm in the Department of Endocrinology and Pediatrics, and I'm in the Translational Genomics of Diabetes lab under the mentorship of Dr. Anna Goin, and we're interested in the genetic causes of diabetes. So the most common causes of diabetes that we often think about are type 1 and type 2 diabetes, and we tend to think of those as polygenic diseases, meaning that there are a number of genes that come together to create your risk for developing the disease. But we're also interested in what's called monogenic diabetes. And that means that there's a single gene that actually causes your diabetes. And this is really important because it can change how the patient treatment is approached. So in this landmark 2004 paper, they actually were able to identify one of these genes, KCNJ11, as a cause for neonatal diabetes, which is diabetes that presents in the first 6 months of age. And knowing this information can actually allow these patients to switch from multiple daily insulin injections, which is the standard treatment, to a more precision medicine treatment, which is just a daily pill that they take. So this is really important. And so it was really fortuitous when one of our collaborators contacted us and let us know that she had a spontaneous mouse develop in her mouse colony that developed neonatal diabetes. So these mice have significant hyperglycemia, which you can see in the upper left, as early as 1 week of age. And they also have what we consider failure to thrive, which is something we also see in human diabetes. We think this is related to a defect in the ability to develop beta cells, or the insulin-producing cells. So that's shown here on the bottom. Our controls showing insulin stained in brown have these nice healthy islets, and our mutants have much less staining. We were able to use whole genome sequencing to identify a single base pair change in the intron of a DNA replication gene called GINS4. And what we think is happening in this in this gene due to this base pair change is actually a defect in splicing. So this base pair change creates a new splice site in intron 4, which leads to a defective mRNA product and a truncated protein, which we predict leads to a loss of function mutation. We also know that there's a lot of overlap between these polygenic forms of diabetes and these monogenic forms. So a lot of the genes that cause monogenic diabetes have less severe defects that can also contribute to your type 1 or type 2 diabetes risk. So we looked at genome-wide association studies to see if this same gene, GINS4, could also be implicated in human diabetes. And we do see, so this is plotting various phenotypes, so cardiovascular in orange, glycemic traits are shown in green, and there's a really high signal for type 2 diabetes. So that suggests that this gene not only has a function in neonatal diabetes, but potentially these polygenic forms of diabetes as well. So my goals for this project, we've got this really interesting mouse and we found this interesting mutation. And so now we're working on understanding exactly how this mutation is leading to diabetes. Again, we think it's probably related to failed beta cell development. So we're using embryonic stem cells and our mouse model to understand exactly where during development this problem comes up. We're also looking more closely at the splicing defect to understand how these changes in protein actually lead to this defect. And then we're further going to investigate the role using human databases of neonatal diabetes to screen for GINS4 mutations, and also further explore the role in type 2 diabetes. So big thanks to my mentor, Anna Goin, all of our lab members, our wonderful collaborators, and all of our funding sources, especially MCHRI. Thank you.
I think we have time for 2 questions.
Yes.
Is there a particular phenotype?
Oh, we'll give you a microphone, Dr. Buckingham.
Is there a particular phenotype in the type 2s who have this gene defect? Has anyone looked at that?
No one's looked at it yet, so it's an exciting, unexplored area for us to look at.
And the DNA replication mutant, is there any other tissues that are affected in the mouse? Like photosensitivity or cancer susceptibility?
That's a great question. So it has been implicated in human cancers, so high expression of this gene, increased risk of liver cancer, pancreatic cancer, and some others. And a full loss of function of this gene actually leads to an embryonic lethal phenotype. So likely this is kind of a partial loss of function phenotype that we're seeing.
Thank you so much. This is really interesting work.
All right, and now for our next speaker, we have Dr. Zoel Quiñónez, who's a clinical associate professor in perioperative and pain medicine in the Department of Anesthesiology.
Good afternoon. Thank you for the opportunity to present what I think is a pretty cool project. I looked at access to pediatric ECMO centers using geospatial analysis and the influence of minority status and poverty on access to pediatric ECMO. So ECMO is extracorporeal membrane oxygenation. It's sort of a mainstay now in the care of children with complex heart disease and children who are in any hospital that's a tertiary quaternary care center. But it's increasingly becoming standard of care. And you can see this in that the American Heart Association recently adopted it as part of its PALS recommendations. Because it is becoming a standard of care measure, it's important to understand the state of access to this technology, to this medical service, and access gaps for the sake of equity and extension of access to a life-saving technology. So we hypothesized that, or the project was to map the access and access points of pediatric ECMO and look at whether there are differences in a couple of sociodemographic characteristics. And we chose minority status and the proportion of the population living under the poverty threshold. So we use the ELSO registry, which is voluntary reporting, but it's the best we have in terms of identifying pediatric ECMO programs. And we use that information to map pediatric ECMO using a platform called ArcGIS, which some of you may be familiar with, the geospatial analysis platform. We defined the catchment area for this technology as 200 miles, and we did so partly in consultation with Kate Ryan, who organized the ECMO transport program at Lucile Packard Children's Hospital, but also with other publicly reported catchment areas from other programs like Johns Hopkins, which also states 200 miles. What you see there on the left is the catchment area for each individual program. What you see on the right is those dissolved boundaries into what would be the catchment area for the continental US. This is an illustration. We did this for Alaska and Hawaii as well as part of this. And then we layered that information with data that's publicly available through the US Census Bureau and available through this data platform, ArcGIS. And we used, as I said, poverty estimates and also minority status as sort of a general sociodemographic factor. And this is what we ended up with in terms of access to pediatric ECMO. And what we found is that 43% of the total US land area does not have access to this, what is becoming standard of care life-saving technology. And that represents about 4.1% of the total US population. In terms of the two sociodemographic factors that I talked about, the population that does not live within the access point is poor, so a higher proportion living under the poverty threshold, and also whiter. So the main findings are that 1 in 25 people live in an area without access to pediatric ECMO based on our criteria. There are certain regions that are more represented in those without access, which is the Rocky Mountains, the Great Plains, Midwest regions, and some of the coasts. The population that lives in that area that lacks access, as I said, tends to be poor, tends to be whiter. There are several limitations to this that are part of subsequent analysis, as this was an initial first look, is that access isn't simply defined by driving distance as we defined it. Increasingly, air transport is becoming a greater mode of transport, at least in some parts of the country, weather dependent and also dependent on the physiologic state of patients. The term minority obviously encompasses a very diverse and heterogeneous population that has different histories of marginalization with regards to access to resources. And so that's also a limitation that a more stratified approach would be appropriate. And also, and that's where you end up with things like that. The population that has less access is wider. This is largely due to population density effect, that populations living outside of high density areas tend to be wider. And if you really want to look at access, we have to look at things like what hospital are you most likely to be served by, and things of that nature. So conclusions, a large portion of the country, at least by land area, lacks access to this technology. Extending ECMO access to places that don't have it is likely going to be an investment in health equity, given that it tends to be a poorer population and one with a lower population density. We obviously need more stratified analysis to really look at access in a more complete way. And there are other factors like motor transportation and population density that we would have to look at as well. Thank you.
We have time for a couple questions. Dr. Shah?
Did you consider using the birth population as the target population rather than the total population?
Yeah, there's actually data, and this is what we're doing now for under 18 on the U.S. Census, which would be a little bit more specific to the project itself. So we're looking at the under 18 population and the birth population in terms of access.
Birth certificates are available to you through CDC. You could actually use the births.
Awesome.
And how would the results differ if we were looking at adult access to ECMO? I'm wondering in how many of those areas without pediatric access is there adult ECMO?
So adult ECMO is more broadly available. I haven't looked at the specific data, but there are papers out there that look at rural versus urban. And obviously urban centers have a higher access to ECMO based on distance to hospitals that have that. It is more broadly available, but it's not something that we looked at here.
Maybe time for one more? Anybody else? All right, thank you.
I'm not sure how to work that.
Our next speaker is Dr. Roshani Sinha, a postdoctoral scholar from stem cell transplantation.
Thank you. Thank you so much for this opportunity. I'm very delighted to be presenting our project, which is developing a novel humanized model of juvenile myelomonocytic leukemia with NF1 loss of function mutations. And we used CRISPR-Cas9 technology, so I'm excited to present it because it's World CRISPR Day today. So JMML is a disease that arises in children. It's a blood cancer, which arises with an increased myelomonocytes as well as monocytes. I don't think I can point. And the median age of diagnosis is around 2 years, and some of the key features or unique features of the disease is that it's a largely extramedullary disease, and there is hypersensitivity to GM-CSF in vitro. It arises with mutations in the RAS pathway genes, of which I'm going to focus on the NF1 mutations where there's loss of function, as it drives one of the worst prognosis in these patients. So NF1 is actually a tumor suppressor gene, and it negatively regulates RAS, which means when it's absent, the active bound GTP RAS goes up, and the downstream pathways are also upregulated. And so this disease is very rare, and it's difficult to treat. And one of the problems is that there's lack of samples because it's rare, or lack of models. So we ended up deciding to develop a model for this using CRISPR-Cas9. So we targeted the exon 2 of the human NF1 gene using CRISPR-Cas9 guides to introduce these loss of function. We used hematopoietic stem and progenitor cells. Since it's a juvenile disease, we used cord blood cells, and we introduced this deletion, after which we confirmed whether we do have NF1 gene knockout or not by different assays. So we first performed Sanger sequencing, and we found that there were more than 75% of cells that did have this indel frequency, and this was consistent with actually putative functional knockout scores. So to confirm whether they actually did functionally behave like JMML patient cells, we performed the CFU assay with colony forming unit in the presence of GM-CSF. So if they are JMML-like cells, they should form more number of colonies compared to controls, which they did. And we saw actually increased number of myeloid colonies as well as erythroid colonies, and the cellularity within these colonies was also more compared to our control samples. We also performed Western blots, and we saw that we did actually knock out the NF1 protein, which you can see on the right blot. So once we were convinced that we do have JMML-like characteristics in these cells in vitro, we wanted to test the leukemia initiation capacity in vivo. For this, the edited cells were transplanted in neonate mice in NSGS-GM3, so they do express the human GM-CSF factor. And we perform the transplant, and then after weaning, which is around 3 weeks, I do weekly blood analysis until week 12, because we don't expect a juvenile disease to go past that stage. What we saw is that all of the NF1 knockout transplant recipients actually had a median survival of 33 days. And the phenotype of all of these mice was that there were no lymphocytes actually produced in them, and a really high percentage of CD13 and 33 positive cells. We also saw a high CD56 expression. So we compared our analysis with what has been published with patient-derived xenografts, and what you can see is we do actually see that we were able to recapitulate the decreased lymphocyte compartment and the increased 13 and 33 compartment that we expect to have. So we're very happy with that, and then we wanted to confirm other aspects of the disease, such as the extramedullary aspect, which is 80% of the disease is found in other organs. So we did see extensive splenomegaly in all of these mice, which is consistent with patients presenting at diagnosis with enlarged spleens. And as the disease progressed, the size of the spleen and the infiltration also seemed to increase. We confirmed this further with histological analysis, where in the NF1 knockout recipients, the red and white bulb regions of the spleen were completely disrupted, and there was extensive infiltration that we found. We also found extensive infiltration in the liver, which is consistent with the findings of JMML patients. As you can see, the human CD45 are the transplanted cells, and we do see them being infiltrated in the entire liver as well as in the lungs. And all of these are consistent with the findings of JMML patients. So with that, we were successful in recapitulating a JMML-like disease by introducing NF1 loss-of-function mutations. And with that, I would like to thank my lab, my PI, Dr. Bertena, all the members of the lab, especially Rachna, who's helped me loads on the project, our collaborator, Dr. Matthew Porteous, without whom this wouldn't be possible, and Rosa Romano, Esmond Lee, BIN's program for cord blood samples, and our funding. And thank you all.
I think we have time for one question. 2 questions. All right, well, I'll ask one. Now that you guys have done this model, what might be your next steps?
So I want to do secondary transplants to see the long-term repopulation capacity, but that's ongoing. And then I'm also looking at some of the genes that we do find in the patient that are differentially regulated. So we're looking into that as well. There's a lot that we can do. And then once we confirm all that, it would be a great model to test therapies. That's something that we want to do as well, because right now there's only stem cell transplant available for these patients, and there's like 50% still have a relapse.
Thank you. Congratulations. This is wonderful work.
Thank you so much.
Oh, one question.
For the great talk, you showed that the wild type mice you showed week 12 and the knockout mice week 5. I'm just wondering how the wild type mice look on week 5 as well.
Yeah.
So you have an idea of the development that's going on when you transplant them.
Yeah, great question. We did actually look at that. I haven't shown it here, but the only difference that we see is there's more B cells, because the T cells do take longer to get engrafted. So I see more B cell compartment. The myeloid compartment is pretty comparable, and then T cells are a little bit fewer because they just take longer. Yeah. Thank you for the question.
And we have our final speaker, who's Dr. Yanming Wu, who's a postdoctoral scholar in maternal fetal medicine in the Department of Obstetrics and Gynecology. Go ahead.
Good afternoon. Thanks for this great opportunity. I'm happy to introduce my work today. I'm studying on a novel in vitro stem cell model to study maternal endothelial function in preeclampsia. So what is preeclampsia? What are the challenging aspects? Preeclampsia is a common pregnancy complication characterized by the new onset of hypertension and proteinuria after 20 weeks of gestation. With greatest morbidity and mortality, it affects 5 to 7% of all pregnancies, impacting 8 million mother and infant pairs worldwide annually. While the cause of preeclampsia is not fully understood, maternal endothelial dysfunction is thought to be a critical component to the preeclampsia pathogenesis. However, studies to dissect maternal endothelial dysfunction is hampered by the access to patient systemic primary endothelial cells. To achieve this, in order to achieve this purpose, we established the endothelial cell model from the protocol as highlighted here. First, maternal peripheral blood was collected and reprogrammed into the induced pluripotent stem cells. Then the cells were further differentiated by activating Wnt signaling pathways, addition of growth factors, and max sorted by the endothelial cell markers. To explore the endothelial function in our established model, cells were exposed to pooled banked sera from normotensive sera, preeclamptic pregnancy, normotensive postpartum as a non-pregnant comparison, as well as 2 control groups, the basal condition and FBS condition. 4 endothelial functional assays, including cellular viability, migration, tube formation, and nitric oxide release, were also evaluated. Among all the assays we tested, levels of nitric oxide release were significantly lower after incubation with preeclamptic sera compared to the FBS. Normotensive and non-pregnant treatment were also lower than the FBS, but higher than the preeclampsia. For tube formation and migration with representative pictures demonstrated on the right side also show that the reduced level in preeclampsia compared to the FBS. The impaired endothelial functions were not attributed to cell death, as no difference was observed in cellular viability among all the treatment groups. So in this project, we are able to establish a cell model to study the maternal endothelial dysfunction in preeclampsia. For the next step, we would like to explore the genotype and phenotype effect by using gene editing techniques. We also want to understand what placenta-derived factors in the sera that contribute to the disease development. Well, big thanks to my supervisor, Dr. Wing, our collaborators, and my lab members, as well as all the foundations for their great support to this project. Thank you.
Thank you. It's wonderful work. Okay, we have some time for some questions. Dr. Oro?
That was a great question, a great presentation. The question is, used one iPS cell, was that a normal person do you know that was it from a normal patient? And
Yeah.
would you predict that if you used iPS cells from mothers that had preeclampsia, would you see a different effect?
That's a very good question. So the current iPSC cells we used come from the normotensive pregnancy background, and we have already recruited multiple iPSC cells from normotensive background right now, and we're currently working on this. Meanwhile, we also collected the iPSC cells from patients with preeclamptic pregnancy background. That is the next step. We will also want to understand how the cell responds to different sera treatment.
Right. So then your data suggests that the cause of preeclampsia is not the endothelial cells, but things that the endothelial cells respond to. Is that correct?
Yes.
There's one more in the back.
My question is sort of related. So if in all of the factors that you mentioned about the causes of preeclampsia, you listed genetic, and I wanted to ask you what the evidence is for potential genetic factors and if there are any gene candidates.
Yeah, right now the data only demonstrate the result from only one patient sample, and we're currently working on to see the endothelial function in other patient samples. So we have some candidate target the preeclampsia associated genetic variants. So for the next step, we plan to screen those variants across different samples and to see using gene editing techniques to knock out this variant to see whether there will be any genotype and phenotype effect. Yeah, that's the next step. Thank you.
Great. Any other questions? All right. I think that concludes our session. I believe our next session isn't until
we will have a break.
We have a break.
We'll come back to 2:05.
Back at 2:05.
Yeah.
All right. Thank you.
Thank you.